start by thanking Stan Fields and the department for putting on what I think is going to be a very interesting interview series, and also for giving me a chance to grill Ben. So, Ben, you've had one of the most amazingly diverse research careers, starting with an undergraduate degree in chemistry and then going through many transitions to your recent work in molecular evolution and systematics. And so I'd like to talk to you tonight about as many of these transitions as we have time for. So to start, let's start with your pre-UW days. Your graduate work and your early work at the University of Illinois was during an incredible period, the early days of molecular biology. Could you tell us something about the work you did at that point?
Well, let me tell you a little bit first about the early days of molecular biology.
Okay.
There was this period where a few people called themselves the RNA Tie Club, and they were concerned with the relationship between genes and proteins. And there was suggestive evidence that RNA had something to do with that, but not very strong evidence. And in those days, Francis Crick was right in the middle of all that. He traveled very widely. This was about 10 years after Watson and Crick. No, not 10 years. Less than 5 years after Watson and Crick's structure. And at the time I was a graduate student at Harvard, Jim Watson had just come to the biology department. And my professor, Paul Doty, who was a physical chemist, macromolecular physical chemist, had a lot of people working on protein structure, some on DNA. And when I came along, he was very eager to find somebody to work on macromolecular properties of RNA molecules. I found out much later on reading Judson's book was because Doty and Paul Zamechnik, who was a leader in protein synthesis at that time, were next door neighbors in Cambridge, and they'd been talking over the back fence. But that wasn't presented to me. So anyway, Crick would come and visit Harvard about twice a year, and he would come to the Doty lab, which was a huge lab. They had about 15 graduate students, most of them working on polypeptides. And he would tell us absolutely everything that was going on in the whole world in what we now consider molecular biology. And he was a very keen observer, and he liked to theorize. And one of my great accomplishments as a graduate student was I proved that one of his theories was wrong. That, in fact, ribosomes didn't each carry an RNA molecule specific to making a protein from that ribosome, that all that RNA in ribosomes was doing something else. Well, that's probably sufficient detail of that part of my life.
Okay, how about let's move on to the University of Illinois, then, to your work there.
Okay, now, first of all, I should explain that I never became a postdoc. It was, as far as my family went, it was not the usual thing to do. Both of my brothers got graduate degrees and got jobs. One as an engineering professor, the other one as a geologist. And I think the postdoctoral period was just becoming fairly routine at that time. The Sputnik went up when I was in graduate school, and the National Defense Education Act followed shortly after that. So there gradually became more money for science. I figured, since I was at that point father of a 1-year-old girl, who incidentally came just in time because my student deferment was up. And I would have been off in the aftermath of the
Ooh.
Korean War had it not been for my daughter Anne.
Ooh, you owe her.
Yeah, so I figured I'd better go and get a job. And Jim Watson had been giving a series of lectures at the University of Illinois. He came back from there and mentioned that they were having a buildup in biophysical chemistry, and he'd put in a good word for me there. So I started out, I had a lab in the physical chemistry division, but I began to make a lot of ties with the people in biochemistry there as well. One of whom just walked in the door, Samir Deeb. And I guess the first summer I was there, I started a little research collaboration with a microbiology professor, Saul Spiegelman, and we did some experiments. Spiegelman used to be very excited about the membrane fraction of bacteria, and he would pulse label them with radioisotopes, and he thought interesting things were happening there. The whole idea of working with radioisotopes, doing metabolic experiments, that was entirely new to me because my training had all been in chemistry. So Spiegelman, my graduate student Mel Green, and I did some work that summer, and then along came Masayasu Nomura, who was a postdoc with Spiegelman and who had spent the summer in Watson's lab at Harvard. He looked at our results and he thought they were sort of total crap. So he took this highly labeled RNA fraction from T4 infected cells and ran it through starch electrophoresis and noticed that the peaks of radioactive RNA and the peaks of ultraviolet absorbing RNA didn't match. This RNA was, and in effect he discovered messenger RNA at that point. That is usually attributed to others, but actually that experiment was done before the ones you read about in textbooks.
The other thing you did at that time, which was critical to all the work we did when I was a graduate student and later in recombinant DNA, was the development of the use of nitrocellulose filters and development of techniques for DNA and DNA RNA hybridization. Could you tell us about that work?
Well, the very same RNA that I was talking about, radioactively labeled after T4 phage infection, seemed to be a very good candidate for this hypothetical messenger RNA that Jacob and Monod were writing about. And I had heard from a contact from the lab where I'd worked before as a graduate student, from Doty's lab, that Julius Marmer there had been able to get the two strands of DNA to come back together and DNA to renature. So I thought, well, maybe this would be a test for complementary sequences. Now, there had been double-stranded polymers made by Alexander Rich, but they were just homopolymers. There wasn't any complicated sequence relationship. So, in fact, I showed that the RNA made after T4 infection was complementary to the strands of T4 DNA. And to do that, I used cesium chloride gradients and took advantage of the fact that RNA molecules have a buoyant density which is high, and they would go to the bottom of the tube unless they were bound to DNA. Now that was a fairly impossible assay technique because you had to spin for 4 days, and you had to drip out all these fractions from the bottom of the tube and count the radioactivity and so forth. So when a visiting Norwegian professor, Agnar Nygaard, came and decided he wanted to work with me for a year, we kind of fooled around with other ways of making a separation between free RNA and RNA that was hybridized to DNA. And actually I knew a little bit about phenol extractions where DNA went to the interface, and I fooled around with that, and it kind of worked. It was very inconvenient. And so we thought, okay, well, the other two-phase organic system often used for extracting DNA is to have a chloroform phase and an aqueous phase, and we'll make some RNA-DNA hybrids with radioactive RNA, shake it up with chloroform, and then sort of capture this hypothetical precipitate at the interface. And we happen to be using nitrocellulose filters. It kind of worked, but it was really noisy. And so Aganar began decreasing the amount of chloroform layer and checking to see if the DNA ended up on, when he poured it through a filter, supposedly collecting the precipitate. And eventually the amount of chloroform went down to 0, and the DNA still stuck. And we found, in fact, double-stranded DNA went through the filter, heat-denatured DNA stuck to the filter. And of course, now there are lots of surfaces that have DNA attached to them in a much more elegant way.
So at Illinois, you were in the Department of Physical Biochemistry, or Physical Chemistry, I think, and then you came to the Genetics Department. That seems like a kind of surprising shift. Could you tell us about how that happened?
Well, how many people here, other than Samir, ever lived in Central Illinois? You did.
Chicago.
Yeah. Well, that part of Central Illinois has trees now, but at the time I was there, they had just had their beautiful campus row of elm trees die of Dutch elm disease. So we used to go to Indiana to see a tree. And the other thing that was a little depressing was when we came to the town, there was a sign that said, "Two good things in Illinois, corn and reliable furniture." So I had worked in the Rockies as a college student. I worked there every summer. I was fond of the West. And so I was very receptive to the idea of moving to a more beautiful part of the country. And I got a message, maybe a phone call from Paul Doty, my mentor, that the University of Washington Chemistry Department was looking for a faculty member. I guess one of their physical chemists had moved to Oregon. They had an opening. I went out, and my faculty host was George Halsey, a guy who's a real character. But he was recruiting me. He was doing a good job, and making sure that I met everybody on campus who had interests possibly related to mine. And I got the impression that where things were going to happen on this campus was in the new building that was on the drawing board for biochemistry and genetics. I was introduced to Herschel Roman and to Hans Neurath. And so I turned down the job in chemistry because I'd really gotten tired of going to seminars on quantum mechanics. Just nothing else all the time, a little bit of chemical kinetics. And felt that I needed to learn some new things. And then a little later I heard from genetics. And I think maybe Stan Gartler had a hand in this somehow. I don't know. But I'd come to the attention of Herschel Roman and came out here and liked the department.
Mm-hmm. So the 60s were a quite amazing period in the Department of Genetics. So Herschel had founded it in 1959. Arno was here, Dave Stadler, Stan, of course. And then shortly before you came, John had come and Larry Sandler. And so that was a pretty small department. But over the next few years, quite a number of important people came. I'm probably going to forget some: Gus, Lee, Walt, Breck, Joe. Tell us about the atmosphere in the department at that point, and how people's research interests affected research, and other, for that matter, interests affected you, affected each other.
Well, I guess from a scientific point of view, we were kind of feeling our way. We had a rather small space in Johnson Hall, and a lot of nice social interactions because we were all working so close together. We had a tiny coffee room where we had journal club. And then we got this beautiful new lab space down on the lower campus, and suddenly became diluted out and split between two floors. And I think that wasn't really good for the intellectual atmosphere until more people were hired, and so I think at the time, around the time Walt Fangman and Lee and Joe came, it became a more serious scientific operation. There definitely was an induction period there.
Mm-hmm.
During that time, we all had a lot of fun, though, playing softball in the summer and going off into the forest and doing various things.
I'm forgetting, what year did the J-Wing open?
65.
65, okay. So in the early 70s, you made one of your pretty dramatic shifts from studying E. coli and its viruses to studying yeast. How did that come about, and what were the were there influences of the department that played a role in that shift?
It's hard to put my finger on exactly what moved me in a particular direction. I think when I first came into the department, because I didn't have any training in genetics, I was a bit of a hired gun for DNA stuff, maybe, and probably expected to become a geneticist, which didn't happen very rapidly. And I think during my sabbatical in University of Geneva in 1968-69, I was in an excellent group where particularly bacterial and phage molecular genetics was going on at a high level. And one could see clearly that the next stages in our science would be to try to understand higher organisms in similar detail and understand how their genes work. I would say the eukaryotic molecular genetics around the University of Washington up till 1970 was very holistic. It was not reductionist in the way that bacterial genetics was. And I kind of began thinking about that and thinking about how I was going to become a biologist and what the right niche for me would be. And that's where, in fact, in 1969, while I was still in Geneva, I decided that I would be looking into yeast and trying to sort of combine the molecular approach with the kind of genetics that other people in the department were doing. And right after that, when I came back, we started working on yeast RNA polymerase.
Oh, so late 60s.
1970.
1970, And then other people joined the lab who became involved in some of those early yeast studies. Richard and Anita. Richard Zinnemer and Anita Hopper.
A few years went by before that really got going. You know, I can remember very clearly that around that time, maybe 69, 70, we had a visit here from Tom Broker, who had just finished his graduate studies at Stanford. I think he worked with Bob Lehman there. And he was telling me about some projects just starting up there in the kind of DNA enzymology that would lead eventually to molecular cloning. And he made a few predictions, all of which turned out to be true. And that gave me the idea that, well, gee, if it's possible to do it, I would like to work directly with some yeast genes, ones for which you already knew the gene product. And that kind of narrowed down to two types of things. One was the cytochrome C structural gene, which from frameshift mutant analysis by Sherman, we knew a good chunk of the DNA sequence, and the nonsense suppressors that were studied in genetics by Don Hawthorne. We knew what their gene product was, transfer RNA molecules. I can also remember meeting Maynard Olson at while I was attending a Gordon conference, and he was faculty member at Dartmouth, and kind of spinning out some of my pipe dreams for him. And Maynard has always been kind of an optimist. He's probably one of the few people who paid much attention to them.
Smart move.
And luckily, I was able to recruit. I think he and Betty had decided they wanted to come spend a sabbatical in Seattle, regardless. And he was looking around to see, well, what is there? I mean, he knew he wanted to look into molecular biology. So I was the recipient of those interests. Mm-hmm. And then he was very important in making all of that happen.
You mentioned a serendipitous result earlier about the nitrocellulose experiments. The other day you told me about a serendipitous result with his first studies of where he was attempting to label tRNA, tyrosine tRNA.
Yes, right. In our region here, the transfer RNA expert was Gordon Tenner, a biochemist at University of British Columbia. And Tenner's laboratory had, and they were among other things working on the transfer RNA genes of Drosophila, and they worked out a very nice, elegant chemistry whereby you could add a reagent whose name I can no longer remember.
I can't help you.
To an amino acid tRNA, and it was the reagent was easily iodinatable by peroxide and I-125, something like that, so that you could make a specific label that was dependent on which amino acid you put on. So it would have a purified amino acid tRNA synthetase, say for alanine, and they would charge it with alanine and then put this reagent onto the alanine and then iodinate it. And so that looked great to us, and so we knew various people, especially Don Hawthorne, had figured out that some of the super suppressor genes, Ocher and Amber suppressor genes, were encoding tyrosine tRNA. There were 8 of them. And we had the idea, well, okay, we'll make a probe for tyrosine tRNA genes following the Tenner technology. What happened, though, was because there's a modified base on the anticodon of tyrosine tRNA, isopentenyl A, the control RNA, which had never been charged with tyrosine, iodinated very beautifully, because isopentenyl was reactive with this reagent. And so that there are some other isopenteno A tRNAs, but this RNA was sufficiently pure that the tyrosine species was getting all the label.
Mm-hmm. I think there have been probably quite a number of surprises that came along in your research over the years, but one that what dates back to that time was the discovery of the intervening sequences in tRNAs, as some which at the time kind of didn't fit what we called the central dogma. How big of a surprise was that? What was your thinking about that at the time?
Well, you know, I think in the summer of 1977, I was at the Woods Hole lab teaching the physiology course, and the sequencing, of course, the whole project was basically Maynard Olson's project, and Howard Goodman was in the lab for a brief time. He had a sabbatical in Kyoto, but he cut it short because he was collaborating with Bill Rutter in San Francisco to clone insulin, and he really felt like he needed to be a little closer to San Francisco to keep his eye on Rutter. So he asked me if would I have a project that he could do quickly. And so he got Maynard into Maxim Gilbert sequencing. So that work was done with the Maxim Gilbert technique, and they found this intervening sequence. And I think I was off in Massachusetts when that happened. So maybe that was why it worked. But you know, intervening sequences in like globin genes and adenovirus, they were all happening about the same time. It was within a year.
Mm-hmm. So lots of surprises.
Yeah.
So your lab played important roles in the early tRNA cloning, cytochrome C, etc. But your lab also was the first one to clone genes by complementation, wasn't it? In yeast, that is.
It could be. I mean, I think the really key discovery for yeast molecular biology was yeast transformation, which Hinnon, Hicks, and Fink figured out how to do. And it just happened that I had made arrangements with Kim Naismith to come and work as a postdoc. And the minute he heard about yeast transformation, he was insistent that he was going to get into that, and he, carefully worked everything out. So, I mean, he was largely responsible for that. And then he collaborated with Steve Reed in Lee Hartwell's lab to clone CDC28 by complementation.
Yeah, he did. I think he did MAT first, didn't he?
No.
CDC28 was
later.
First. Okay.
CDC28, and then with Ted Young and Valerie Williamson to clone ADC1, alcohol dehydrogenase.
Okay. So,
I mean, the other thing I think where Kim excelled was he made a very good library in a 2-micron vector, and there were a bunch of people trying to do that at the time, and he was way more efficient than Jim Broach.
Yes, I used that library for many years. So, after years of lots of innovative basic research and covering a number of areas, you ended up expressing the hepatitis B S gene, surface antigen gene in yeast. I'm wondering whether, was this a brilliant idea of yours? Hey, maybe we could make a hepatitis vaccine in yeast, or if not, how did those experiments develop?
Well, you've already established pretty convincingly that I don't do things that way. I just kind of stumble along and hope something will turn up. So the real motivation for Gustav Ammerer's work was to better understand how yeast RNA polymerase II started making an RNA, what directed it to start in a certain place, and to transcribe it actively. And in order to study that, he had made a fusion between the CYC1 coding region and the upstream part of ADC1, and then just to make sure that nothing about the CYC1 was providing directions for starting an RNA, he said, okay, well, I'm going to take something else. He tried, first of all, rat growth hormone, which he got a cDNA clone from an Austrian friend of his who was working in San Francisco. And that seemed, and the RNA was made, and the protein assay was negative, and many years later, he became convinced that the RIA kit was no good.
Oh, no.
So that may well have worked.
Oh.
So anyway, Gustav and I both felt, well, let's collaborate with some people who have a protein they really can assay for reliably. They know what they're doing, and the assay is good. And I hadn't made a visit to Genentech the year before, and so I got in touch with Ron Hitzman down there and Dave Goodell, and we worked out a collaboration just to answer that question about, what's the starting signal for yeast RNA polymerase II. And then it, suddenly dawned on me and other people that this had value. And there was momentary excitement about the fact that it made interferon, which was pretty misdirected because interferon did not have all the properties that were ascribed to it. But I very quickly thereafter heard from Bill Rutter, who had been working with Howard Goodman. Howard Goodman had cloned the surface antigen gene of hepatitis B virus, and then he moved to Mass General Hospital, and Rutter's group did some experiments trying to express that protein in E. coli, and they were not successful. And so Rutter, with Merck's backing and two other groups, wanted to avail themselves of our technology. And so then I became practically oriented. But I didn't plan it that way.
So around the same time as you were doing that work, you also became chair of the department, following Herschel, which does not sound like an easy job. And you also started Zymogenetics, at that time called Zymos, with Mike Smith and Earl Davies. How did the department and the university feel about your biotechnology work and getting involved in the start of a biotech company?
Oh, the answer to that is very complicated. So the switch over from Herschel being chairman to my chairmanship was in the summer of 1980, and the expression of polypeptides was the following winter in February. The company formation was talked about in the summer of 1981, and it happened in 1982. So as far as the university being the chairman of the department, I was receiving some signals from provost, deans, associate deans that really didn't filter down to other faculty, because that really had been the model in genetics, that Herschel took care of all that stuff and didn't want the faculty to be distracted. So I guess I kind of continued that. I mean, the faculty didn't want to be distracted. But the moment the word got out about this company, it was a bad scene. I I think I talked about that mostly with Gus Dorman, who tried to be kind of intermediary between faculty members who were concerned that it was going to be unfair or they were going to be dragged into something they didn't like. Mm. As far as the university administration, they love that sort of thing. It's just like today, where they want to turn the university district into a high-tech campus, and they've got all sorts of plans. But that was just starting then, and they were really eager to get into technology transfer. But I don't know, it was in retrospect, I probably should have taken time off from the university, certainly from being chairman, if I wanted to pursue that company. Because by the time I'd been in it a year and a half, I'd had enough, and I discontinued my affiliation. Although I'm happy to say that Zymogenetics did perfect a process for making insulin in yeast, and that's what many diabetics receive in this country today.
So looking back on that period and what's happened since, what are your thoughts about academics being involved in industry?
It causes industry to be much more successful. I mean, I felt that up until there were biotech companies, if you had friends who went off and got a job at a pharmaceutical company, you never heard anything about them. They didn't publish papers, and not much happened, with a few exceptions. And companies like Genentech, and to some extent Cetus, they set a new standard because they were able to capture that enthusiasm and really do very innovative things. And I think and I hope universities will continue to be a cauldron of innovation as they have been in the past. We all hope there will be research grants, right?
Okay, I'm skipping over a few years now, but the most recent and I would say quite dramatic shift in your research has been to molecular evolution and systematics, and a shift to organisms such as plants and red algae. I was looking at some of your papers, and I actually have to read this. They made mentions of organisms such as alveolates and straminopiles. I don't even know how to pronounce them, let alone what they are. How did this?
Let's just call them protists.
Okay. How did this research shift come about?
Well, I mean, time and again in our discussion here, I've mentioned people who came into my lab and made things happen. The person involved in that is John Stiller, who was a graduate student in the Department of Botany, worked on algae with Bob Walland, and he and I had talked. I actually knew him while he was a graduate student. In fact, he discovered that red algal ribosomal RNA or DNA molecules had introns. He was co-discoverer of that as a graduate student. So he was interested in using the molecules I was familiar with as sort of metrics for molecular evolution. And since I was just entering a different field, it wasn't that easy to find, well, not at all easy to find people as good as John Stiller.
You told me that it's hard to find the thread in your research over the years, but I actually do see a thread, and that's studies of RNA, transcription, regulation of transcription, the polymerases, and then using the, in your molecular evolution studies, you've been using RNA polymerase 2 subunit genes in that work. Don't you feel like there's, even with all the diversity, don't you feel like there's been a clear thread to your research?
Well, that's true. RNA is where it started because basically nothing was known about the solution structure of RNA. That's why my advisor put me on it. And tomorrow morning, Dale Lindsley and I are going to go to a rhododendron garden, collect a bunch of rhododendron seeds for growing up to make RNA. To do RNA-seq, which is the important information you need to annotate a genome sequence. So yeah.
What's the most exciting result you've ever had?
Oh, I would say the hybridization of RNA to phage DNA. Because of the really slow analysis, you know, there was lots and lots of time to think about, you know, what's going to happen. And it was very hard to find the proper conditions. I mean, it's kind of laughable when you think about it from today's perspective. But I was really obsessed with the worry about the competing reaction of the two strands of DNA wanting to go back together as opposed to the RNA. Of course, I didn't know at the time that the RNA-DNA complex had a higher TM than the double-stranded DNA. One thing that I duplicated from Julius Marmer's original protocol for DNA renaturation. He slow-cooled denatured DNA. It originally slow-cooled in an autoclave. He was trying to kill the transforming activity of DNA, and he heated it to 110 degrees and took it out and had much more residual activity than if he put it in a water bath at 100. And figured out that it's because when you turn off an autoclave, it cools slowly. So I got this huge water bath that was, I don't know, I think it was originally a Coke cooler, and would put the tube in there at 85 degrees and then just turn it off and let the temperature slowly. So that was a smart move, because regardless of what the optimum temperature for renaturation is, at some point I would hit it for a hybrid formation. I'm happy to say I've learned the same principles used in PCR these days.
I remember reading your hepatitis B paper with Gustav around the time it came out, and thinking how amazing it must have been to see those particles that looked like the virus-like particles that are found in chronic hepatitis B patients in the yeast cytoplasm. Wasn't that an amazing result?
Half the people who worked on that at another lab at Genentech maintained that it was expected to happen. I wasn't expecting it to happen. So, yeah, I mean, I think the instant you saw those particles, you knew it was going to be a good immunogen, because it was so similar to something they had already developed from carrier plasma.
Okay, before turning things over to questions, I'm going to ask kind of a broader question relating to the field as well as your own work. And the first time I read Eighth Day of Creation was towards the end of graduate school, which was late 1970s, and which was obviously a very exciting period in the field with the early days of cloning. But reading that book, it just struck me in the 20 years difference between the start of our research careers. I mean, what an amazing period of time you'd been involved in, and you know, heard about this basically going from understanding nothing, almost nothing about molecular biology, not learning about the structure of DNA, finally accepting that DNA was the genetic material, blah blah blah, messenger RNA, genetic code. What have been the most exciting periods in the field, both for you and the field as a whole?
Well, I think the time from 57, 58 until the mid-60s, a lot of things happened. Genetic code was cracked. Messenger RNA was, as a concept, was established. That was a very exciting period, and I was fortunate to have a couple of things that related to that. I'm still very proud of the fact that I found the ribosomal RNA and found that it was a rather homogeneous substance and was not the message. That was mentioned in Judson's book, but no mention of whether any human being was involved in doing it. And the hybridization of messenger RNA. So I had my big toe in that. You know, I talked quite a bit about the late 70s, early 80s, certainly being able to isolate genes and study them and sequence them and so forth. That was a second high point, and I would say the genomic era is the third one. And with the rhododendron genome, which mostly is being done by Jay Shanduri's lab, but we're involved in to some extent, I have a little bit of contact with that as well. But it's just, when you think of how crude the techniques were when I started, and how we always worked with massive quantities of stuff, and now the microfuge tubes just keep getting smaller and smaller and smaller. And people talk about sequencing one DNA molecule. It's a big change.
Oh, would have been unimaginable. Okay, well, let's turn things over to questions, and I'll try and remember to repeat the questions.
Tell us about the future, Janel. Tell us about the future.
Yeah,
Good.
I thought a little bit about that. I just hope it's not systems biology. Well, you know, when you read Science Magazine or today's Tuesday New York Times science section, you see all these strange organisms from wherever. So maybe there'll be a flowering of that kind of natural history. I mean, we've all done our thing with model organisms, and perhaps people will get tired of that and become naturalists again, with many more tools at their disposal.
Ben, talk a little bit about your teaching that you did in the department. I have personal memories of both being a TA for you and also taking your wonderful graduate class. So tell us a little bit about what you felt was important to get across to students and how you mastered genetics.
I'm not sure that ever happened, actually. How did I master genetics? Well, I guess it was for about 20 years I taught a winter quarter course in what we would call molecular genetics. And as I look back, the part I enjoyed talking about transposable elements in maize. I don't know whether I did that the whole 20 years, but...
Not for us, I don't think. No.
Oh, I guess toward the end. Now in rhododendrons, we just find them all over the place. Yeah, yeah. I taught I can't remember what the number was, but the general genetics course I taught for several years.
371, I think.
And I first taught biology maybe before that, and then with Ann Paul I taught Bio 210 or 200, whatever it was called. Enjoyed doing that for several years. Yeah, I was teaching that 200 level course when the Gustav's expression and yeast experiments were done. I remember that.
I actually have a story to tell. I don't know if either Bonnie or Ben remembers this. The first time I TA'd, the first time Bonnie TA'd, was the first time you taught the whole genetics course. Herschel had taught the classical genetics part previously, and you taught the molecular part, and you had taken over the whole class. And I remember one day you were, I guess this was using transparencies at that point, and you were talking about the egg and the sperm would fuse to form the zygote and so on, and you said, and the zygote has, this was humans you were talking about, the zygote has 34 chromosomes. And I looked at Bonnie and I said, what's he talking about? And then suddenly I realized, that's yeast. We always would talk about yeast having 17 chromosomes. We never talked about the diploid number. So I'm sitting there thinking, do I correct him? All these students are writing this down. And I decided I'd better. Ben, I think you're confusing yeast and humans. So that's my favorite story from Ben's teaching days. Other questions?
I have a question. Oh, I'm sorry. No, this just, when Ben spoke about the company, it brought back a memory, which I don't have all the details on, that Herschel, who was always interested in the stock market, he really thought he'd buy some shares in this company. And so I remember he said he didn't have the name completely right, but he remembered Zymo was part of it. And so they bought him shares in some totally different company, which had the word Zymo in it.
Yeah, that other company was going to sue for infringement.
Oh, there was, I see that.
And it stood for zirconium metal oxide semiconductors.
That's why it's Zymogenetics instead of Zymos, right?
That's when the name was changed.
Yeah.
Ben, you trained in chemistry as a graduate student, and yet you've made transitions into many other fields. Was there anything special about your training that helped you to make those transitions, to be fearless and just switch into something new?
It might have been family influence, because my father was a mammalian systematist. My mother had done work in botany before she started having sons. And I had a brother who was a geologist, and he used to tell me really interesting stories about that. Right now I'm really fascinated by geology and glaciology and stuff like that. I think it's a natural history background. Now I think graduate training for most people is going to narrow their focus. And certainly mine did narrow for a while. But I think I get frustrated if I'm doing the same thing for too many years. It's nice to run into a new set of ideas, a new set of problems.
I have actually two questions, but I'll ask one first. I've heard a rumor that the reason you got into rhododendrons was that you were impressed or envious of Herschel Roman's rhododendron garden at his house. Is that true?
I was impressed by it, yeah. I was impressed by his lovely setting there. And
I mean, the
I
story
I
was
thought-
that you then just started growing them at your
Well,
house.
he had a direct hand in me getting a lot of rhododendrons. He had a buddy named Halfdan Lem, and Lem was a retired fishing boat skipper, a Norwegian who was about 6'8", big old guy, who was a very successful hybridizer of rhododendrons. And when nursery was going out of business, Herschel told me about it, and he said, go out to Aurora Avenue and get a pickup or a truck or something, and you'll get a good deal on these big rhododendrons. So I did, and planted them in my garden, and they ignored them for about the next 30 years. But the real scientific interest in rhododendrons is in the wild species, which I wish I could go to New Guinea and China and everywhere and pursue some more, but I find that it's not hard to find students who want to do that. What was your second question?
The second question was, I just want to take you back to your PhD work on ribosomal RNA. Were you the first person to see ribosomal RNA separate into large and small subunits? And if so, are you the one responsible for us calling them 18 and 28 sRNA?
Well, the answer to the latter part is yes. And there were people in Watson's lab. There was a very active ribosome group working on E. coli ribosomes, especially Alfred Teissier, a Swiss biochemist. And they kind of figured out the two ribosomal subunits, the 30S and 50S. And as it turned out, one of the RNA components that I saw was from the 50S, one from the 30S. But I didn't show that in my thesis. I just showed that there were these two. And S stands for Svedberg, and that's a Swede who had a magnificent oil turbine centrifuge he invented, and so they named the unit after him.
Any other questions?
Then last year you gave a really nice seminar about what happened after the hepatitis B surface antigen was actually turned into an actual immunogen and used. That must have felt really good. I don't remember all of the numbers, maybe you don't either, but how many kids were immunized and how many kids never got Hep B? Has that sort of been a capstone? I would think it might have been.
Well, it certainly was. I mean, I had a lot of fun looking up all that public health data and trying to understand the cellular immunology of it, which is quite interesting and complex. I think the best measure of how successful it was is that in many of these societies, which had a high incidence before of people who are carriers, it's gone down tenfold, or maybe sometimes more than tenfold, to a few percent. There seem to be a few people on whom the vaccine doesn't work. Some of those are successfully immunized if you use a little bit longer version of the S protein, like the kind that's in the mature virus particle. But in some case, it seems that some HLA genotypes don't respond to this vaccine. And I'm actually wondering whether that has something to do with, I don't know much about, different countries, different ethnic groups and their HLA types, but this may be also involved in why the incidence is high in some places relative to others. That's the part of the preparation I didn't complete.
Okay, any other questions? Okay, well, I think
Thank
that's
you all
it.
for coming. You were